What is experimental research? Experiments allow researchers to study cause and effect relationships, although not perfectly. This method is the gold standard, the preferred option, but it requires a lot more resources than descriptive and correlational studies. A true experiment has 4 essential characteristics. 1. The participants are randomly assigned to different groups. This means that every participant in the study has an equal chance of being selected to join the experimental group. We often use computer programs to help us with this. Without random assignment, it's technically a quasi-experiment. Second, the researcher creates a control group that will be used for comparison purposes. This group will be treated the same as the experimental group, except for the manipulation under investigation. Third, in one group, the researcher manipulates what is called an independent variable. And fourth, the researcher measures the effects of that manipulation on another variable called the dependent variable. Imagine we are investigating whether a new training program for college students improves their grade on a group project. We divide our class into 2 groups, the experimental group and the control group, and then we randomly assign each person to one group. The only difference between the groups will be the independent variable, whether they attend the training or not. At the end of the semester, we will measure the dependent variable, which is their grade on the project, and compare the 2 groups. If we find statistically significant differences between their grades, it suggests the training program had something to do with it. To determine if a difference between 2 groups is meaningful and represents real-life differences, we consider what is called statistical significance. Essentially, this is the probability that the group differences we observed could have happened by chance. If the significance is less than 0.05, it means the group differences are probably not due to chance, less than 5 times out of 100. Instead, they are due, at least in part, to the training. If the significance is more than 0.05, it indicates the group differences are probably due to chance, more than 5 times out of 100. In other words, the group differences aren't all that meaningful because they're just as likely to happen in the natural course of the world and have little to do with the training program. Of course, this calculation is not absolute. It's only a probability. This is one reason why it's important for the research community to test hypotheses and theories again and again to hone in on the true probabilities of relevant phenomena. Let's define some other relevant concepts before moving on the last part of this section. Researchers commonly measure subject variables to determine whether the independent variable affected different groups of people in the same way or differently. These variables are unique to individuals and cannot be manipulated. For example, we may want to compare the grades of different races, age groups, or genders. A confounding variable is the official name of any extraneous variables that could have influenced the relationship between the independent and dependent variables. In every experiment, there are plenty of variables that researchers cannot control. The temperature of the room, the brightness of the lighting, the weather outside, the mood of their participants. All these variables and millions more can influence the outcome of research, but it isn't practical to measure them. When researchers design their experiments, they consider internal and external validity. Internal validity is a measure of how confident we can be that our manipulation of the independent variable is the actual cause of the change we observed in the dependent variable. High internal validity indicates that our manipulation is the cause of the change. It is negatively affected by confounding variables and experimenter expectancy effects, which occur when the researcher's expectations about the study influence the participants' behaviors. To enhance internal validity, researchers use control groups, blind conditions where even the researchers don't know who is in which condition, and random assignment. External validity represents how confident we are that the results can be generalized to other people and other situations. How well do the results represent what the population experiences? The generalizability of the results can be threatened if we use a non-representative sample, like a convenience sample, or if the research setting is too unfamiliar, irrelevant, or unrealistic. To enhance external validity, researchers make it a priority to collect data from representative samples. Instead of relying on first-year college students in need of research credit, more and more researchers are inviting employees and other sectors of the public to participate via the internet. They also try to create realistic research settings that match the real-world situations as closely as possible. One way of increasing experimental realism is to use deception or hire confederates to play certain roles. The challenge with these strategies is they may serve as confounding variables and have an unintentional effect on the outcomes of the study. In the next part of this lecture, you will explore the final research method, the meta-analysis,